right, so this page is going to deal with osmosis, which is the diffusion of water. So if you remember, diffusion is moving particles from high to low concentration. It is passive transport. It doesn't require any energy. So osmosis is going to be the same thing, only it's dealing with water, moving from an area of high concentration to an area of low concentration. So looking specifically at animal cell osmosis, in the notes we looked at red blood cells. And for hypotonic, I always emphasize the O because the red blood cells swell up and look like a big round O. Right, so I'm just going to draw a couple little O's here. And we know that water is moving into these cells, okay? So there's more sugar, salt, or starch on the inside of those cells, which means there's less water on the inside of those cells. So water is moving in, and eventually that cell is going to explode. In an isotonic solution, everything is balanced. So I'm going to just kind of draw a red blood cell, nice and normal, has that little indentation in it. So just a group of red blood cells, normal. Water is moving into and out of the cells at a normal rate. Everything here is balanced. I should probably write something over here. So water moves in. Okay, and then for our last one, hypertonic. These ones, they have more water on the inside of their cells. Ooh, those are terrible. More water in the inside of their cells, so the sugar, salt, and starch is on the outside. So it's going to start to pull the water out of those cells. So water moves out. Okay, so the cell is going to shrivel up and eventually it's going to die. So plant cell osmosis is a little different, I guess. So we know that plant cells have cell walls. So these ones are just going to be a little bit bulgy in the cell wall. And let's say I'll draw orange will be my cell membrane. So if green is my cell wall, my cell membrane is going to be pretty bulgy too. Same thing that's happening is that water is moving inside of the cell. Eventually that cell is going to burst as well. So same thing that's happening in the red blood cell for animal cell osmosis. For my isotonic solution, it's going to look nice and normal. No bulging. Cell membrane looks good. Everything looks great here. And I have water moving in and out of the cell at the same rate. My hypertonic, my cell wall is going to look somewhat normal-ish, but this is where we see the difference. On the inside, that cell membrane is going to be shriveled up because my water is moving out of that cell. Okay, so remember that sugar, salt, and starch pulls water to itself. So wherever the water, wherever that stuff is, the triple S's, the water will go. So let's change my pen color here. In which condition has the cell been placed inside of a sugar solution? So inside of a sugar solution, it would be hypertonic. There would be more sugar outside of the cell, so it's going to pull the water out of the cell, making the cell shrivel up. In which condition does the cell have too much sugar inside of it? Okay, so there's more sugar on the inside than there is on the outside, which means it's going to pull water in, and that would be a hypotonic solution. And in which condition has the animal cell reached homeostasis? isotonic is nice and balanced, and that's where we're going to have homeostasis. So look over the animal cell osmosis pictures at the top and answer this question. Which picture would show the cells of a freshwater fish, okay, a freshwater fish, if it were placed inside of a saltwater aquarium? Okay, so freshwater fish. I'm going to draw a fish. Pray for me, y'all. This is going to be bad. Okay, here's my... That's terrible. I'm so sorry. Here's my freshwater fish, and it is going into an aquarium. Let's make some little trees. Okay, and what is inside of that aquarium? I'll pick dark blue, I guess. A whole bunch of salt. So that means that there is going to be more salt, okay, outside of the fish. So what's going to happen if there's more salt outside of the fish? That water is going to pull the water outside of the fish's cells. So it's going to move the water out of the cells of the fish. And it's going to act like a hypertonic solution. It will pull the water out of the cells of the fish. Typo. Which would in turn dehydrate the fish. And fish would be no more. All right, if you have any questions about that, please